Hey everyone, we're doing immune system part 2 today, and the main things we'll talk about are active and passive immunity. We'll talk about vaccines and also allergic reactions and anaphylactic shock, and then autoimmune disorders. We don't really go over those so much in the notes, but it is in the pre-lecture assignment. So if you look at our question of the day, what is a vaccine made of? Or I think a better description of that is what do they put in a vaccine to make it effective? All right, for the pre-lecture, I am going to go over this first table. It kind of compares and contrasts active immunity versus passive immunity. So the first row here is, does the individual produce the antibodies or are they given the antibodies? So with active immunity, of course, they produce. So just remember with active immunity, it's your body actively doing something. They have to actively produce the antibodies. Whereas with passive immunity, the antibodies are given to the individual in a variety of ways. Next row, is it long-lasting or temporary? Active immunity is long-lasting. Whereas passive immunity is temporary. Because over time, those antibodies will break down. So an example of active immunity would be like getting a vaccine that causes your body to produce the antibodies, and passive immunity, it can go through the placenta, the breast milk, or something called antibody injections, where they actually inject specific antibodies into a person. So for the second part of the pre-lecture assignment, it goes over autoimmune diseases. It mentions two, rheumatoid arthritis and lupus. So for the exam, just make sure you know that if you were given like a definition that you could say which one is which. All right, active immunity, we will start with that. This is when antibodies are made against an antigen. And remember, an antigen is just like a marker on either a cell or something that's in the body. And active immunity is facilitated by the B and T cells. What is wrong with my pens? There we go. And this occurs either after a pathogen infection or after immunization. And I don't know if this bothers you guys or not, but my printer ran out of colored ink. So I'm sorry this is in black and white. It's much more effective and easier to teach when it's in color to point out certain colors, but I will get a refill soon. So we're already to our question of the day. What is the vaccine made of? So I'm not going to sit here for a few minutes. You can pause it, but pause the video for a minute and think, what could you put in a vaccine that would cause an immune response, but not get the patient sick? So you want something to be recognized as foreign, but not get the patient sick. So pause it and think about it. Okay, now we're back, right? Um, so the main three you need to know is the first is called live attenuated. This pen's gonna die too. This is not my day. Live attenuated, basically, this is when pathogens or the disease causing agents are weakened so that they reproduce very poorly once inside the body and cannot cause disease, but they can induce an immune response. So I'm just going to say weakened here. And I'm going to draw a cute little virus. I'm going to give him some antigens. This looks like something out of a nightmare. And I'm going to do a frowny face because he's weak. So your body can still recognize these antigens, but it can't make you sick. The second one is a killed pathogen. So we're going to draw our same guy. I might have to switch to a regular pen. I hope you can still see this. Yeah, it should be good. There's antigens going on here. But this one's dead. And these pathogens are completely inactivated or killed with a chemical, so it cannot possibly reproduce inside your body or cause disease, but it can evoke an immune response because it still has those antigens there that your body can create antibodies for. And then the third is a subunit. And with this one, you're just putting parts of the pathogen. So I'm drawing some of the antigens there. The vaccines are composed of certain proteins, the antigens, that reside on the surface of the pathogen, and the fragment is responsible for the immune response in the host. So these are the three you need to know. And if you're familiar with the COVID vaccine, that's an mRNA vaccine. You don't necessarily need to know that, but if you want to do more research into that, you can look up mRNA vaccines. So a little more about vaccines. They expose the immune system to the antigen to generate those memory B and T cells. And we'll talk about this chart below in a second, but the first exposure, or the first shot, if you will, leads to, I'm saying too much, I'm sorry, a small increase in antibodies. And it's a short-lived response. It doesn't protect you for very long. The second exposure, which they usually call a booster, leads to a high increase in antibodies. And the results are longer lasting. So if we look at this table here, on the y-axis, it's showing antibody concentration from low to high, and over here it's showing the time in days. So the primary response, or the first exposure, or the first shot, is going to increase the amount of antibodies for a small amount of time, and then it decreases. with that secondary response or the booster shot, you see you get a really quick increase in antibodies and they last longer in your body. So to be the most effective, you need, I guess depending on the vaccine, but you're going to need that initial shot and then later on a booster. Which is what they did with the COVID vaccine. And we're not going to get into that argument. I know it's kind of a hot topic, not so much lately, but I was first in line to get the first shot, also first in line to get the second shot, and I haven't gotten a booster since, which I should probably do, but I just haven't because I ended up getting COVID anyway. Okay, just disregard anything I just said. Um, so vaccines are given during all stages of life, and some vaccines will eventually wear off, so a booster is necessary. Again, I'm sorry that I don't have a color copy of this, but this is the schedule for children, and it shows you all the things they vaccinate for and at what age they do it. And then let's see, when you would need like your second dose. So you don't need to memorize any of this by any means, but it is interesting to see. And then for adults, there's one here as well. And where's the flu? Influenza. So everyone through age 19 through 65 or older should get one dose annually. And the reason for that, if you don't already know, is because the influenza virus mutates every year. And so you need to get a booster for the new variations. Let's see, what else do I want to point out here? Do you guys see a vaccine for the common cold, the rhinovirus? I do not see one because there isn't one. And the reason for that is the rhinovirus mutates so quickly that you'd have to be getting vaccines all the time. So instead, we just deal with the symptoms of it, and it's usually not deadly. So no vaccine for that. Why does it take so long to develop a vaccine? So if you look at this, and the reason that we put this on here is because the COVID and the rush for the vaccine. So, and I wish I had a chart. There is one. I should have printed it. But just to give you an idea, it wasn't until 1980 that smallpox was eradicated. We still don't have a universal flu vaccine. That is something they're working on. And it took until 1994, which was probably before some of you guys were born, but after I was born, for polio to be eradicated in the US. And there were some problems with the polio vaccine. There were some bad batches that actually ended up giving kids polio. So vaccines are highly regulated. So, how a new vaccine is developed, the FDA has three phases. Phase one, and you can read through this. I know you can't see it on my copy probably, but you can look at the different phases, and then they make sure it's safe and blah blah blah, and then they can put it out there. And I think that was a lot of the concern with people and the COVID vaccine is that it came out so quickly, whereas it took us from like I don't know when polio started to become widespread, but clear up until 1994. So, if I gave you the idea earlier that I am against vaccines or the COVID vaccines, that's absolutely not true. I was just irritated that I still got COVID anyway, but I understand the science behind it, so it's fine. Okay, on to passive immunity. This is when the individual is given antibodies directly. And remember, this is temporary. The results are not long lasting. Because the antibodies will eventually break down. And you lose immunity. So, like we talked about in the pre-lecture, antibodies are passed to offspring through placenta and breast milk. And this is helpful because the baby is likely to get the same pathogens as the mom because they're in the same environment and in very close contact. And so for the mom to pass those antibodies to the baby is helpful. There is also antibody injection, like you see here, when they're actually injected into the patient. And they'll do this if there's an accidental exposure of a healthcare worker to some pathogen. My computer just updated, so it keeps popping up with things. And an example of this is a gamma globulin shot. And I don't know a lot about that. That's probably something I should look up. Or they can inject antibodies from someone who already had the virus or even from an animal. And something else they can do for passive immunity is to use cytotoxic T cells. Remember, those are like the killer cells, and they can actually be engineered in a lab to target cancer cells. All right, so on to hypersensitivity reactions. So hyper means high, so like higher than normal reactions. These are instances where the immune system responds and it actually causes harm to the body. So 3 examples of this are allergies, tissue rejection after some sort of transplant, and then autoimmune disorders. Again, we went over 2 in the pre-lecture assignment. So just to take some little notes here, what happens with this is the allergen attaches to mast cell receptors. So for example, the mucus cells of your nose, and then the mast cells release histamine, and histamine will bind to the body cells, and this induces the allergic response. So you've probably seen something like this before where they will test different allergens to see if you're allergic to them with a skin allergy test. And if it swells up like that, you're having a release of histamine. And so that's how they can tell if you are allergic to something. Again, the autoimmune disorders, that's going to be in the pre-lecture assignment, so just know what those two are that we went over. So we'll talk about allergies. This is when the immune system attacks substances that the body normally would not. And it responds to allergens. Some examples are pet hair, pollen, and some foods. So what cells release histamines? Those are going to be the mast cells. And the antibody on the mast cell surface binds to the allergen and causes that release of histamines. And so what this will do as far as allergies goes is it will cause increased mucus within mucous membranes. And this leads to the watery eyes and runny nose that you often get with like seasonal allergies to pollen and things like that. An asthma attack. This is caused by a release of histamine specifically in the airways. And this is going to cause them to constrict, to get more narrow. And that prevents the air from getting in as easily. AIR, sorry about that. Let's rewrite that better. And onto our last page, anaphylactic shock. This is when an allergen enters the bloodstream and then it can become systemic or spread throughout the entire body. So this is often caused by food allergies. So I will have students every year that have like severe peanut allergies or allergies like that to certain foods to where they have to carry an EpiPen, which we'll get to. And I have to be trained how to use it, and for some reason it freaks me out. I've never had to do it, but let's hope that day never comes. Bee stings. A lot of people are highly allergic to bee stings. Sometimes injections, like vaccines. You'll hear of stories of people that have a bad reaction to an injection or a vaccine. And also some medications. So what causes this severe reaction is your arterioles will dilate. And remember, dilate means to get bigger. And if you increase the the diameter of the arterioles and you still have the same amount of blood going through, that's going to decrease the blood pressure. So they'll have a big drop in blood pressure, and then it also causes restriction of air passageways. So they have difficulty breathing. So to help counteract this, they use an EpiPen. And what the EpiPen has in it is epinephrine, which is where the epi comes from, which is basically adrenaline. And this is the same as the fight or flight mechanism. So if you remember back to our sympathetic versus parasympathetic nervous system, the sympathetic uses epinephrine as the, what's the word, neurotransmitter. And so what that is going to do is we want it to do the opposite of this. So it's going to constrict the arterioles. So if you make them smaller and you have the same amount of blood pumping through, that's going to increase the blood pressure, bring it back up. And opening of air passageways. And as far as I know, and you guys might know about this, more about this than I do, I don't have any allergies, nor do my kids or anyone in my family, so I've never had to deal with this besides at work with my students. But from what I understand, the response to the EpiPen is pretty quick. It's pretty effective and works pretty well. Okay, tissue rejection is when the immune system attacks transplanted tissue. So if someone gets a kidney, a heart, or a liver transplant, the immune system will often try to attack it. And the foreign organ triggers an immune response. So the cytotoxic, those killer T cells, attack it. And so when they do this, the organs are carefully matched for antigens. Curving around there. Because we don't want the recipient's body to recognize that as foreign so it doesn't get attacked by those cytotoxic T cells. So they use immunosuppressive drugs. Examples are cyclosporin and I don't know how to say this one, tacrolimus. And what those two drugs do is they inhibit T cell cloning. So what that will do is make it so no cytotoxic T cells are made, so they can't attack the organ. And I think this next one is super interesting. Xenotransplantation. This is when you get an organ transplanted from an animal. And they can actually genetically engineer pigs to grow organs that lack antigens. So your body, there's nothing to recognize as foreign because there aren't antigens, which is really cool. So that is it for immunity, and we have one more packet on this module. It is the infectious disease, and then we're going to be on to exam 4. Like I said, we're just flying through this. I feel like we just had our exam and now we're ready for the next one almost. I think it starts, I need to check the syllabus, but I think it starts on the 15th. So I will get that last lecture uploaded as soon as I can, and then we can go over the study guide and get started on that 4th exam. So thanks for hanging out, and I hope you guys have a great day, night, morning,